Hello my dear students, welcome to our channel Rajesh Biology and I am Rajesh MSc Zoology. Today our topic is gametogenesis, in that spermatogenesis. First of all, what is gametogenesis means? Gamete. There is two types of gametes are there. That is what? Male gametes and female gametes in the reproductive system. Gametogenesis. Genesis means generating or producing. So, genesis of gametes, producing of the gametes. How the gametes are produced, that process is called gametogenesis. Gametogenesis means what? It is the production of the gametes from the primary sex organs of the male and female reproductive system. If it is the male reproductive system, the gametes are called sperms. The gametes are called what? Sperms. So, the formation of sperms is called what? Spermatogenesis. Spermatogenesis. If it is the female reproductive system, if it is female reproductive system, the gametes are secondary oocytes. Oocyte or ova. So the formation of ova is called oogenesis. This formation of ova is called what? Oogenesis. So in this video, we are going to talk about spermatogenesis. That means from the male reproductive system. That too particularly from the primary sex organ. That is testis. From the testis, sperm cells are formed. How the sperm cells are formed? From the testis, where they exactly formed from? So, just revise once. What are the testes? Testes are the male primary sex organs. They are oval in shape, covered by tunical bugynia that inwards and divide the testis into the testicular lobules, 250 lobules, and every lobule contains seminiferous tubules. So, we are taking one seminiferous tubule out. Just revise the seminiferous tubule structure also. Seminiferous tubule internally lined by two types of cells. They are germinal epithelium and Sertoli cells. Germinal epithelial cells helps in the form of sperm cells, and Sertoli cells provide nourishment. So this is what we completed in the last class. Now from here we are taking the germinal epithelium along with the Sertoli cells out. So I am taking this part of the seminiferous tubule, I am pasting here. Okay. So this is what? Wall of the seminiferous tubule. This is what? Wall of the seminiferous tubule. In this wall, the wall is lined by two types of cells. One is elongated nourishment cell, nurse cell, that is called what? Sertoli cell. This is called Sertoli cell, which is going to provide the nutrition to the germ cells, to the spermatozoa also. And now I am drawing these mother cells. Mother cells are the germ cells which are going to produce nutrition, which are going to provide nutrition to the male germ cell. This is not nutrition. Male germ cells are going to produce sperm cells. Clear it is now? Now, I just want to tell you one thing. These are the mother cells, are also called stem cells. Spermatogonial mother cells or stem cells. Stem cells are mother cells. And how they behave? They want to increase their number. Before they are going to produce the spermatozoa, they want to increase their number. They want to continue their legacy in the seminiferous tubules. How they continue their life? First, they increase their number by the mitotic divisions. For example, 50 stem cells are present. Just for example, 50 stem cells are present. If every stem cell form the spermatozoa, if every stem cell form spermatozoa, what will happen after 20 ejaculations or after 50 ejaculations? All the stem cells becomes expired and there is no stem cells to produce further spermatozoa. But it is not happen in this our society. A human male can produce spermatozoa up to the death. The sperm formation continues till the death. How it is possible? How it is possible means these sperm cells, these mother cells first increase their number, they multiply their number. Okay, like if there is 50, they become 100. Out of 100, 50 participate in the spermatogenesis process. Only 50 participate in the spermatogenesis process. The remaining 50 continue their normal, regular spermatogonial mother cell life. Okay, when they want to produce the sperm cells again for the next ejaculation, at that time also they increase their number, they become 100, and 50 of them follow the spermatogenesis process. It is just like an investing process of the so many businessmen persons. So if they get 100 rupees profit, they are going to invest only 50 rupees. The remaining 50 rupees profit they will save in the saving accounts. When they get the 100 rupees from that 50, again they are going to invest 50% of the profit. So like that, spermatogonial mother cells want to continue their life by showing the mitotic divisions. So spermatogonial mother cells, first what? They multiply in number. They multiply in number. How they multiply? By the mitotic divisions. By the mitotic divisions. So if it is the spermatogonial mother cells, these are also spermatogonial mother cells which are newly formed. So spermatogonial mother cells, how they form? They are formed by the mitotic divisions. Clear it is first, that is the mitotic phase. Okay, now, so this newly formed spermatogonial mother cells, they grow in size and they become the primary spermatocytes. They grow in size and they become what? Primary spermatocytes. They become large and they are ready to form primary spermatocytes. Primary spermatocyte. And the Sertoli cell is elongated like this. This is the Sertoli cell elongated like this. So what are these cells? These cells are primary spermatocytes. Primary spermatocytes, which are actually participate in the spermatogenesis process. Which are actually participate in the spermatogenesis process is what? Primary spermatocyte. Up to the primary spermatocyte stage, there is no change in the genetic information. There is no change in the genetic information. Why? They are just show the mitotic divisions. There is no meiotic division. They become multiply. They proliferate. They increase in their number and in their size. So spermatogonial mother cells which are diploid, which have 46 chromosomes, and newly formed spermatogonium. This is actually spermatogonium, not the mother cell. It is the spermatogonium. It is also diploid and 46 chromosomes. Mother cells increase their number. 50% of the mother cells which are newly formed, they are considered as the spermatogonium, not the stem cells. That spermatogonium developed or grown into primary spermatocytes. So primary spermatocytes also diploid in number, diploid, and 46 chromosomes are present. Now the process of spermatogenesis is going to start now. And this process is called what? Spermatocytogenesis. Of course, it is not mentioned in the NCERT or in the board. But if you want to know this process, you can know like this: spermatocytogenesis. Spermatocytogenesis is the process of formation of primary spermatocytes. Process of formation of primary spermatocytes is called what? Spermatocytogenesis. It is a part of spermatogenesis. So now I'll erase this and I'll start with the primary spermatocytes and I'll explain in a schematic presentation. Okay? Primary, okay, first we will draw the spermatogonial cell, spermatogonium, which is decided to form the sperm cells. Spermatogonium. Spermatogonium is developed into the primary spermatocyte. What it is called? Primary spermatocyte. Primary spermatocyte. And these both are 2n in stage. 2n in stage. Now the process of meiosis begins. So primary spermatocyte undergo the first meiotic division to form haploid secondary spermatocytes. Haploid secondary spermatocytes. Secondary spermatocytes, which are in haploid 1n in stage. 1n. So this is what? Meiosis 1. Meiosis 1. Primary spermatocytes undergo the first meiotic division and form two equal haploid secondary spermatocytes. Haploid means single n. Diploid means 2n. Okay. So if it has 46 chromosomes, it has 23. Clear it is now? Each secondary spermatocyte continue the meiotic division. I think you may already know what is the cell cycle means. Meiosis has two stages. First, meiosis 1. Meiosis 1 is called reductional division. Reductional division means 46 is divided into 23. Second meiosis division is equal to the mitotic division. That's why meiosis 2 is called equational division, equal division. So the 23 is formed into what? They are called spermatids. They are called what? Spermatids. I'll give another color for the spermatid. It is spermatid. Spermatid. 23. Don't think as it is the mitosis. It is not a mitosis. It is what? Meiosis 2, which look like the mitosis. Meiosis 2. So here also 23 haploid. They are called spermatids. They are called what? Spermatids. So primary spermatocytes undergo first meiotic division and form the two secondary spermatocytes. And the secondary spermatocyte, each secondary spermatocyte undergo the second meiotic division and form how many? From each secondary spermatocyte two secondary spermatocytes, 4. 4 spermatids are formed. Spermatids are also 1n in stage. So this is called what? It is also meiosis 2. Okay. Now spermatids not undergo any division. There is no mitotic division. There is no meiotic division. There is no mitotic and there is no meiotic divisions. Spermatids simply transform. They just transform into the spermatozoa. How they transform? They are become like this. Later they form one acrosome on the head and they become the they form the mitochondria in one part. Okay, the centrioles move towards the two ends and the distal centriole comes out like this, and this plasma membrane cover this structure. Okay, axial filament structure is covered by this plasma membrane. So like this, they just change their shape by forming the different structures. They change their shape. This process is called, it is actually transformation. What it is called? Transformation. You know, in TikTok, so many artists show the different types of transformations. That type of transformation, the person is not going to change. He just change the color of his shirt or something. So just like that, this spermatid change the shape and the structure of its cell. So it becomes like this. This is called spermatozoa. This is called what? Spermatozoa. Spermatozoan. Okay. So like this, spermatozoa is formed by the transformation. Now I'll give names to each and every stage. Okay. Spermatogonium converted into the primary spermatocyte. This process is called spermatocytogenesis. Spermatocytogenesis. There is no problem even though you forget this word because it is not mentioned in the PU level. Okay, or intermediate level, it is not there. Primary spermatocyte undergo first meiotic division and second meiotic division. They form the spermatids that transform into the spermatozoa. This entire process is called, entire process means what? Formation of haploid. It is also haploid. Formation of haploid spermatozoa from the diploid primary spermatocyte is called spermatogenesis. Spermatogenesis is the process of formation of haploid sperm cells. Haploid sperms from diploid spermatogonia. From diploid spermatogonium is called what? Spermatogenesis. Very important. What is spermatogenesis? Spermatogenesis is the process of formation of haploid spermatozoa from the diploid spermatogonium is called spermatogenesis. Now, what is this transformation is called? Transformation have another name. This transformation is called spermiogenesis. Very important. Every time students get confused. The difference between the spermiogenesis and spermatogenesis. Spermatogenesis is complete process. Spermiogenesis is only this transformation. So what is spermiogenesis means? Spermiogenesis is the process of transformation of spermatid into the spermatozoa. This transformation is called what? Spermiogenesis. How many spermatozoa are formed from the primary spermatocyte? Total 4 spermatozoa is formed. 4 spermatozoa is formed. Okay. How many spermatozoa are formed from each spermatid? Only one. Only one spermatozoa is formed from one spermatid. How many spermatozoa is formed from each secondary spermatocyte? From each secondary spermatocyte, only 2 spermatozoa are formed. How many spermatozoa is formed from a single spermatogonium? From a single spermatogonium also 4 are formed. Because it is not like this. 2 primary spermatocytes are not there. Okay. The spermatogonium is grown. It becomes bigger in size and that becomes a spermatocyte. Okay. This is also growth. So like this, there is a few phases. One is multiplication phase. Spermatogonial mother cells multiply in number. Next, growth phase. Some spermatogonial cells growth into they grown into primary spermatocyte. Okay. Next, meiotic division. Meiosis phase. Primary spermatocyte undergo first meiotic division, secondary spermatocyte undergo second meiotic division and form spermatids. This is meiotic phase. And last but not least, spermiogenesis, transformation of spermatid into the spermatozoa. This is called spermiogenesis. Clear it is now? Now I am going to draw the same diagram which I arranged before here, and I am going to explain what happened in the seminiferous tubules. So this is the seminiferous tubules, and it is what? It is the Sertoli cell, and primary spermatocytes are present here, spermatogonial mother cells, and they are transformed into the primary spermatocyte. This primary spermatocyte forms secondary spermatocytes. This secondary spermatocyte form the spermatid, and the spermatid is transformed into the spermatozoa. This is called what? Spermatozoa. So if you observe, all these processes, all these steps are involved, embedded inside the Sertoli cell. Sertoli cell provide nourishment to all the stages. But in our NCERT, it gives Sertoli cell provide nourishment to the germ cells. In our academic books, in like Telugu academy books, it shows it gives the nourishment to the germ cells and spermatozoa. Okay, so whatever it is, at the site of spermatozoa also, the head of spermatozoa is get embedded in the Sertoli cell. It is not get released. Okay, so it is still present in the Sertoli cell. After taking sufficient amount of nourishment, once the sperm cell stomach is full with the food, it get released. It is released from the Sertoli cell. So this release from the Sertoli cell leads to the release from the seminiferous tubules. So for example, I am showing this. This is the spermatocyte. Spermatocyte is embedded in the wall of the seminiferous tubule, actually in the Sertoli cell like this. So it is what? This, my hand is the tube. Which tube it is? It is a seminiferous tubule. Okay. Once it is released, it is comes out from the seminiferous tubules into the testosterone. Okay, like this. From here it is a testosterone, epididymis, vas differentia. So releasing of sperm cells from the seminiferous tubules to out, releasing of sperm cells from the seminiferous tubules is called spermiation. Very important. Spermiation. So here 3 words you have to differentiate. Spermatogenesis, the process of formation of haploid sperm cells from the diploid spermatogonia is called spermatogenesis. Spermiogenesis, transformation of spermatid into spermatozoa is called spermiogenesis transformation. Spermiation, releasing of sperm cells from the seminiferous tubules, releasing of sperm cells from the Sertoli cells. This release is called spermiation. Just like ovulation, here it is the spermiation, releasing. Okay, so these are the 3 words: spermatogenesis, spermiogenesis, and spermiation. Now, how this entire process is controlled? Okay, what is the hormonal control about the spermatogenesis? Now I am going to discuss about that. Hormonal control at the age of puberty. At the age of puberty means it is vary from person to person. It is average. The average age is 13 years to 15 years. Between the 13 to 15 years age. On an average only it is 13 to 15 years age at the age of puberty. The hypothalamus release a hormone called gonadotropin releasing hormone. What it is? Gonadotropin releasing hormone. Previously we may not discuss in the online classes, but hypothalamus is called master controller. Pituitary gland is master gland, and that is also under control of hypothalamus. So hypothalamus is called master controller. How it controls? It releases only 2 types of hormones, either releasing hormone or inhibiting hormone. So here hypothalamus releases which hormone? Hypothalamus releases gonadotropin releasing hormone. Please release the gonadotropins. Like that it gives the order to the pituitary gland. So pituitary gland just receiving the order, okay boss, and I'm going to secrete the gonadotropins. Like that pituitary gland listen the voice of the hypothalamus. So hypothalamus send a order, release the gonadotropins, gonadotropin releasing hormone. And pituitary gland by receiving this hormone, which pituitary? Anterior pituitary. Anterior pituitary by receiving this hormone, anterior pituitary release the gonadotropins. So don't confuse. Gonadotropins are not secreted by the hypothalamus. Gonadotropins secreted by the pituitary gland. Okay, what are the gonadotropins? Only 2 gonadotropins are there. That is LH and FSH. So these 2 hormones work on gonads. Gonad means what? Testis and ovary are called gonads. Organs which produce gametes are called gonads. Testis in male is a gonad. Ovary in female is a gonad. So tropin means stimulating. Hormones which stimulate the gonads are called gonadotropins. So LH in male it is called ICSH. Why it is called ICSH? Interstitial cell stimulating hormone. Interstitial cell stimulating hormone. So if it is the seminiferous tubules, the space between the seminiferous tubule is called interstitial space. The cells which present here are called interstitial cells. So Leydig cells are called interstitial cells. LH is going to stimulate what? Leydig cells. Okay. So Leydig cells going to secrete what? Leydig cells release androgens. Andro means androecium, gynoecium. Andro, wherever you hear the word andro, means male. Generating. So androgens which generate the male characters. Androgens are released, and in that androgen, chief androgen is what? Testosterone. It is a steroidal hormone. Testosterone, a steroidal hormone which is released from the testes. That is called testosterone. And this testosterone is going to help in the formation of spermatogenesis. Again, this process is because of testosterone hormone. When it releases, it is released by the Leydig cells. When it gets the stimulation from LH. LH is released from anterior pituitary when it receives the hormone from hypothalamus. Okay. When hypothalamus releases, hypothalamus knows everything about the body. And when the certain age is reached, hypothalamus gets stimulated. Okay, this is the age to release gonadotropin releasing hormone. And gonadotropin releasing hormone released by the hypothalamus that reaches the pituitary. Pituitary releases gonadotropins, LH and FSH. LH stimulates the testes. Testes release testosterone. Testosterone stimulates all this process. Okay, then about what is FSH? FSH feels very sad because we are not going to discuss about the FSH. Now FSH is happy. FSH means follicle stimulating hormone. And this is going to stimulate Sertoli cells. It is going to stimulate what? Sertoli cells. Sertoli cells function is what? It's going to secrete nourishment to the sperm cells. So what happened? When a person reaches the puberty, hypothalamus recognizes the age and it releases gonadotropin releasing hormone. By receiving the order, anterior pituitary releases gonadotropins. That gonadotropins stimulate the gonad. And the testosterone is going to release. And the nourishment is released from the Sertoli cells. So when this process begins, it requires some nourishment. Every stage requires some food for the division. Okay, without food they cannot divide. So the simultaneously Sertoli cells provide nutrition and the testosterone stimulate the spermatocytogenesis. So spermatocytogenesis, spermiogenesis and spermiation that leads to the ejaculation. This is about spermatogenesis and hormonal control. In next class we will discuss about the structure of the spermatozoa and later oogenesis and ovum structure. Okay, thank you.